Hi, welcome everyone. Thank you for coming. My name is Cheryl Elkin. I'm with QIAGEN Digital Insights, and I'm pleased to be here today with Dr. Ravi Kolhe from Augusta University. The format of our presentation today is going to be a fireside chat. I apologize, there's no fire, but you know, that's actually probably a good thing, where we will explore various aspects of implementation in a laboratory of comprehensive genomic profiling for cancer. So Dr. Kolhe, could you please introduce yourself and tell us a little bit about your institution and the kind of work that you're doing there?
Thank you, Cheryl, and I'm glad to be here. This is my 8th consecutive year at CGC. I'm Ravi Kolhe, Professor and Chair of Pathology at Medical College, Georgia. I used to do a lot of things, but these days mostly administration and molecular pathology testing and signing out.
So we're really interested to hear your perspectives on the full process of implementation of NGS for comprehensive genomic profiling, from sort of panel development and design through interpretation and reporting. So what are some of the trends that you see, starting maybe with panel design, and then we can move on to the reporting?
Sure. So I think at this moment in time, especially in the clinical NGS, the NGS part is getting very mature, but if you go 10 years, maybe 12 years back when we started, we're struggling with small panels, 15, 20 genes, smaller instruments, and then we moved on to, I would say, mid-sized panel, 100, 200 genes. Then the TMB biomarkers came, which needed a bigger 2 megabase panels, and we moved to that. And now I think there's a pretty significant move towards whole exome, backbone type panels, and obviously there's always whole genome sequencing in horizon.
So what benefits do you see from using a whole exome or a whole genome backbone?
I think we are tired of chasing the biomarkers. I mean, every single year there's one or two clinically very significant, helpful for patients biomarker gets added. And even if IVD or even LDT panels take a pretty significant amount of work in the lab to develop, to validate, verify, and if that biomarker is not part of that panel, then suddenly you have to move to another panel. And I think we are sort of tired of this chasing, and that's why most of these labs are trying to see and feel if a whole exome might be a suitable one to avoid this chasing.
So with the whole exome and the whole genome, are you reporting on all of those genes, or do you use a bioinformatic filtering, or how are you managing with all of those genes on those panels?
So there are multiple ways people are doing it. At least what we are trying to achieve is a 1,000 gene panel with 1,500x coverage, and then you have a 100 to 200x backbone for copy number alteration. And then you can get all your TMB, HRD, MSI type biomarkers from the whole exome and have a focus sign out from, I've seen from 600 to 1,500 genes, depending on what you feel comfortable. I've seen people doing smaller panels in a very specific phenotype base. So prostate will have this, breast will have this, brain will have this. So there are multiple ways people are doing it, but I think there is a significant move towards whole exome.
So with a panel of 1,000 genes that you're reporting, what are some of the challenges that you face from the interpretation side?
A lot. I mean, we moved from single gene to whole exome now, and it used to be smaller panel, very targeted approach, and now suddenly you get this pretty significant amount of data to look through, significant filtering. The initial screening is taking a lot more time. On the good side, I mean, the sequencing is getting a lot more cheaper, faster, automation is there, but the analysis and reporting definitely is adding a lot of time to the lab.
So do you use a decision support application in your lab? And if so, how did you go about selecting this decision support?
Yeah, I mean, you have to look at it a solution both on the wet bench and the dry bench side, which is pretty comprehensive, and something which is panel agnostic. So let's say, at least I can give you our examples. We started with 2 main panels. One is 500 gene panel, and another one is another 500 gene IBD panel. And if you're stuck with one analysis and decision support team, then it becomes very challenging to find another solution for another panel. So one of the things we do in my lab is what we do is Kolhe 10. It's an exercise of 10 things we go through when we, each and every time when we try to find a panel or a reporting solution. And one of the most important things is obviously it has to be regulatory compliance. You got your AMP, CAP, ACGME based annotation of the variants. Second and most important aspect is it needs to be panel agnostic. And I think that's what a lot of people are looking for. And third and most important for personally is my time. I don't want to be sitting on a screen and trying to chase information in a bioinformatics system. I want that to be comprehensively presented. There are certain things we look on a metric side, both on the run as well as bioinformatics. And then I want that visualization to be done quickly, perfectly, consistently on every single case I do. And we have in the past compared, I would say, 10 plus different solutions and ended up the one we are doing it, I would say, since 4 or 5 years, which has really helped with the last aspect I am presenting is it cut down my time. There's a pretty significant information I can give it to my patients, and it really is panel agnostic. So we moved from 170 genes to 500 genes, so we're going to whole exome, and now we're testing the waters with whole genome. And if I could keep the exact same decision reporting tree or a bioinformatics solution, I think that makes my life a lot more easy.
Can you tell us a little bit more about the solution, so you use QIAGEN Clinical Insights. Can you tell us a little bit more about that solution and how you use it and how you decided to use it?
Sure. I mean, we got involved with QCI when QCI was bare bones, and we have worked with them, and I think one of the greatest advantage is the integration of N-of-1 with QCI. Now the product QCI1 makes a pretty significant difference because N-of-1 itself was, I would say, one of the major knowledge base and annotation for comments for somatic tumors. And with that integration, the analysis and the primary analysis, secondary analysis, tertiary reporting has become extremely pleasurable, easy, and we could, on the front end, because it's a panel agnostic, we could set it up for any panel we would like, and then push through the exact same QCI and use the last piece of N-of-1 knowledge base to sign out these cases. So it has been a long process, but I think since post-pandemic, I think the product has matured enough that I could comfortably sign out most, if not all, the cases in a relative time. Doesn't matter if it's a 500 gene panel or whole exome, and we are also looking at whole genome now on QCI.
How does the software help you when you have really large numbers of mutations? Like when you're looking at a case that has hundreds of mutations, how does it help you? Can you describe the process that you would go through?
Sure. I think the key thing is, irrespective of what bioinformatics solutions you use, is the validation filtering. So use your validation cohort and pretty strong and robust filters, right from allele frequencies to what the read depth, you have to come up with your own validation filters. And once all those filtration is in place consistently, you have to challenge yourself and make sure that you're not missing anything out. But that initial filtering makes a pretty significant difference, especially if you're going for a bigger panel or whole exome or whole genome. And then you look at each and every variant class separately. So you look at SNV separately, indel separately, amplification separately, deletions, LOH separately, and then come up with your own method of madness so that when you're looking at it, irrespective of what informatics solutions you're using, and then with this different subclassifications of variant class has helped me personally to make sure that I'm not making any mistakes in, let's say, you can't have one standard filter for all the variant class. So our SNV filters, our LODs are different from copy number alterations. LOH calls, deletion duplication calls are different. So you really have to do that homework and then pick up your sign out solution, and I think this is where QCI1 made at least our life and my lab's life relatively seamless now, to the extent that we are looking at whole exome and whole genome purely because of this process in place.
And do you have a way that you can look at this list of variants and the system presents to you initially this list where you can see clearly what are the predicted pathogenic variants and which are the VUSs? Are you able to, is it fast and easy to look at those and make determinations and determine if you agree?
Absolutely. I think not only you can have that visualization, but even once you visualize that, if you want to go in depth on looking at what that particular variant or more information on that variant, the IGV integration to look at that particular variant, the population frequency, the allele frequency, the germline predisposition, everything is in one screen. I don't know how you guys do it, but it really helps me if I want to dive deeper into one particular variant and then try to make sure that, and then you have this opportunity, which again is a part of your sign out, is you can change the tiers. These are not very fixed tiers. And then you can push and prove not only for your lab, but also see what other labs are doing for different, changing the tiers for particular variants.
And you feel it's transparent in terms of the evidence that was used to make the computed classifications and the evidentiary support from the
Yeah, I think this is one where we always struggle with is how do you make a particular variant is benign, likely benign versus pathogenic. And I've educated a lot of our oncologists on how we think a particular variant, it might be a tier 1 because of the clinical reasons, but biologically it might be benign or likely benign. And I think having that clear description, and not only description, but knowing yourself how you're going to classify these variants and then going back to your question, it's presented very transparent, not only presented, but you have an opportunity to add to that discussion or even change that classification before you sign out, I think is a pretty added value.
Right. Is there anything else you'd like to add before we close?
I mean, I know we are sitting on a commercial station, but we have done, and I think everyone should do a pretty significant not only validation, but work to figure out what works for best for them and your lab, because there is, the panels are increasing, the biomarkers are increasing, and then once we slowly start moving towards, I would say, whole genome, it is going to be extremely critical what you use. The filtering solution, the available evidence for supporting your decisions, what can go in the actual report, what can go on the first page of the report. So making yourself comfortable with the sign-out solution is very critical, and then educating the other side. I mean, even including patients in that discussion has really helped us to not only finalize a solution, and been using QCI for the last 6-7 years. And then other thing I would say is you have a great team of people that are extremely open for feedback. We have made significant changes in the template or visualization. That really helps us to be comfortable with the solution and not something which is pushed down on us, and we don't have much leeway in making changes. So the final piece is really enjoyable, and I really hope the next type of whole exome and whole genome fits in with what the 500, 600 gene panels we're doing on.
Thank you so much for your time today, and thank you very much for your attention. And feel free to stop by our booth if you would like to chat further. Thank you.
Thank you.